papillomavirus infection. Bit of a basic virology. It's from the Papoviridae family. It's a DNA virus. Double-stranded DNA virus with protein capsid infected human squamous metaplastic epithelial cells. There are 130 genetically distinct human papillomavirus types, of which 30 to 40 primarily infect lower anogenital region. So it's a sexually transmitted infection. And also there are benign form and also malignant transformation is there. Bit about the life cycle. 6 early E genes governs functions early in the viral life cycle. So that's denoted by E. 1, 2, 3, 4, 5, 6, like. It includes DNA maintenance, replication, and transcription, expressed in low epithelial layers once infected. 2 late genes denoted by L1 and L2 major, that is L1 minor L2 capsid proteins expressed in superficial layers, required to form a new infectious viral particles. HPV gene expression occurs in synochrony with epithelial differentiation. So that's going with simultaneously. So about life cycle, it's a non-lytic virus. So it's not lyse the cells. Infectiousness depends on normal desquamation of infected epithelial cells. New infection is initiated when L1 and L2 capsid proteins bind to epithelial basement membrane. You can see here, permits entry of HPV viral particles into new hair host cells. So that's how it goes on. So from this infection from 0 weeks up to 6 to 12 weeks, it's slowly progressive disease. So you can see viral infects of primitive basal keratinocyte. Then, so these early fragments involved, then virus and cell replicates together, even E2, viral DNA amplification in non-dividing cells, and viral genome at thousands per cells, E6, E7, E1, E2, E5, like. Then virus laden cells ready for desquamation and infection of naive individuals, 1, L1, and 2 are there. So you can see dividing cells, differentiating cells, virus particle assemble. So that's their life cycle continues. Risk factors. Early age of sexual intercourse, multiple sexual partners, poor immune functions, poor hygiene, all depends. Types of HPV viruses. Based on their oncogenicity and strength associated with cervical cancers, we classify them as high risk: 16, 18, 31, 45, 35, 50. Persistent HPV then leading to oncogenicity. Type 6P in invasive carcinoma in cervical intraepithelial neoplasia, 2 and 3 stages. So increased tendency to persist, while low risk types like 6 and 11, it leads to genital warts, subclinical human papillomavirus infection, and laryngeal papillomatosis. About the transmission, it's following the direct contact, usually sexual contact with gentle skin, mucous membranes, or body fluids. Often from the either warts or from subclinical HPV infections. HPV cervical infection is generally limited to women who have experienced penetrative sexual intercourse. Infects epithelial tissue through microabrasions that expose segments of basement membrane. In most cases, infection is multiple, involving more than one lower reproductive tract sites. You can see multiple lesions. So then transmission other non-genital warts like from the formities, doorknobs, tables, likewise touching area. Less common oral to genital and hand to genital transmission. Congenital, conjunctival, laryngeal, vulval, or perineal warts present at birth or that appears within 1 to 3 years of birth is likely perinatal exposure to maternal HPV. Pathogenesis of HPV infection. Persistent infection with high-risk HPV types affects the cell growth by the following mechanisms. HPV integrates into the host DNA. Overexpression of viral proteins E6 and E7. Viral proteins activates cyclins A and E. permitting further cell proliferation. Also mediates and degrade pro-apoptotic gene that's called BAX inhibits apoptosis. So programmed cell death will be inhibited. So there are just about these flow charts like HPV E6 and E7. E6 which binds to P53 while E7 P53 50, P21 reticuloblastoma gene. So E6 activates E680 PT53 degradation and unregulated cell cycle. Immortalization, that is, cells will not die. Similarly, here E7 cell cycle productions. So all continues and ultimately leads to immortalization. Similar thing, don't worry. So here it is E6, P53 binding, P53 binding, it's P53 activation, P53 degradation. So it is where E7 an oncogene product of one of human papilloma virus, which goes and binds to retinoblastoma. So usually promote on cell begins mitosis. So that's where it's bind to this area and leading to the immortalization or inhibit the apoptosis. So there are major risk factors like what early age of coitus, multiple sexual partners, likewise, then HPV infection, epithelial cells change in basal cells of squamous epithelium following exposure. So there are low risk types, you know already 6 and 11, high risk types 16, 18 mainly. So episomal infection is from the low risk varieties leading to condyloma acuminata. So it's a benign lesions and leading to regression. While high risk types 1 integrated into those basement or epithelial cells, it leading to cervical intraepithelial neoplasia grade 1. Persistent of HPV infection is lead to scene 2 and 3, and it leading to invasive cervical cancer. Outcome of HPV infection. One could be either condyloma and latency, and or maybe subclinical infection. And later intraepithelial neoplasia, invasive carcinoma, or otherwise spontaneous clearance. Latent infection. Cells are infected but HPV remain quiescent, so that remain silent. Viral genome is intact, no detectable tissue defect. Production of HPV infection. Completion of viral life cycle increases in viruses. Oncogenes are expressed in very low levels. Both in males and females produce either genital forms, that is condyloma acuminata, or subclinical infection. Subclinical infection may be indirectly identified in cytology as LC. Neoplastic HPV infection, other hand, when the cervical intraepithelial neoplasia grade 3 or cutaneous or cancerous lesion, circular genome breaks up open, integrates DNA at random locations in host chromosomes. So when the transcription is unrestricted of E6 and E7 oncoproteins, those are interfere with functions of tumor suppressor genes. Infected cell vulnerable to malignant transformation by loss of cell cycle control, accumulation of nuclear mutations. Degree of abnormal epithelial mutations used to break lesions histologically as mild, moderate, or severe dysplasia or cervical intraepithelial neoplasia. Bit of condyloma acuminata or acuminatum, also known as anogenital modes. It's a benign papillary lesion or the squamous epithelium. It is associated contact transmission. There are multiple forms soft votive masses. Common site anus, perineum, vaginal wall, and vulva. Caused by male HPV 6 and 11. Microscopically tree-like proliferation of stratified squamous epithelium marked acanthosis, hyperkeratosis, parakeratosis, perinuclear vacuolation, coilocytes formation. You can see it's a histopathology. So it's usually regress spontaneous, but there are autophilos, imiquimod like this we can use topically as treatment. We usually do diagnose by clinical suspicion of the lesion of appearance and cytology, histology, and colposcopy. HPV testing is done cervical cancer screening between more than 30 years. Triage or surveillance of abnormal cytology results, post-treatment surveillance. Detection of HPV DNA, polymerase chain reaction, PCR. There are other commercial RNA probes, Cervista HPV, HR test, southern blotting, and hybrid capture. If cervical cytology and HPV tests are negative, combined in combination, negative predictive value is very high, so that means we can exclude the HPV infection. HPV testing is used as an adjunct test in evaluating abnormal cervical cytology. So that's a part of the cervical screening. In evaluating atypical squamous cells of unknown significance, ASCUS. HPV triage strategy can detect cervical inflammatory neoplasia stage 2 and 3 lesions effectively. Includes pap smear test. Atypical smear after excluding infection. HPV testing, positive for high-risk HPV. Pap smear, then HPV testing, then colposcopy. It's a kind of biopsy. So usually if it is negative, repeat smear after 1 year, those who are high-risk category. Prevention. What is HPV vaccine? It's usually given for 12 and 18 years between. So it's part of our now extended program of immunization. It's a highly immunogenic vaccine, synthetic production of L1 capsid protein of each HPV. So there are different formula of different vaccines. Gardasil is quadrivalent vaccine which covers high risk 16, 18 and low risk 6 and 11. While Cervarix it's a bivalent 2 types 16 and 18. Usually gives 3 doses in 6 months. Effective for at least 1.5 years. Then use of condoms prevent HPV transmission through body fluids and across mucosal surfaces. Behavioral interventions, sexual abstinence, delayed coitate, and limiting the number of sexual partners. Maintain local hygiene to treat vaginal infection. Male partner should be maintained penile hygiene. So it may be reserved high risk HPV infection. Quiet smoking.